Hi, this is Dr. Lauren Launen, and this is the second part of a lecture set on translation. So, we talked about the structure and function of ribosomes in the previous video lecture, and now I want to talk about the stages of translation. So the three stages of translation are the initiation stage, that's the first one, it's the beginning. The second stage is called the extension, or also the elongation stage. So that's actually like doing the act of translation. And then the final stage is just called termination. So let's start, of course, at the beginning with the initiation of translation. So this is all about base pairing, attaching the messenger RNA to the small subunit of the ribosome. So we looked at the structure of intact ribosomes, and you should remember that a ribosome is made of a large and a small subunit. Those large and small subunits only associate with one another during the act of translation. The rest of the time they're actually separated. So what we're seeing here right now in green, this green blob is a small subunit. And so here we're seeing a molecule of ribosomal RNA. So it is embedded within the small subunit, and it is 16S rRNA. So what we're looking at is a photo or a cartoon of a prokaryotic ribosome. This would, however, be very similar to what one would see if you were looking at a eukaryotic small subunit. So don't be distracted by that. So this is actually part of the ribosome itself, of the small subunit, and it's the 16S ribosomal RNA molecule embedded right in there. And it has a region of nucleotides that will base pair with this messenger RNA molecule. Okay, so this is the messenger RNA. It's not part of the ribosome. It's just attaching to the ribosome. This green and this blue here are the actual small subunit of the ribosome itself. And so this sequence right here, we've referred to previously as the ribosome binding site, or RBS. In bacteria, it's also called Shine-Dalgarno. But this is the ribosome binding site right here, and it is binding to the ribosome by base pairing with the 16S rRNA molecule. In the eukaryotic world, there's a sequence that's called the Kozak sequence on their messenger RNA. And so their ribosome binding site is called a Kozak sequence. And it'll function very similarly. So you get initiation beginning with base pairing between the messenger RNA at the 5' ribosome binding site to the small subunit, and it's base pairing specifically with a molecule of rRNA that's within that small subunit. Okay, so the coloring's a little bit different, but the figure works. Okay, here's the small subunit. Here's messenger RNA, and what has already happened is that base pairing through the ribosome binding site in this messenger RNA molecule has happened with a 16S rRNA molecule that is within this brown small ribosomal subunit. Okay, now what? We've moved to the next part of the initiation of translation. And what we've got is a very unique transfer RNA molecule, and it has attached to the area called the start codon. And the start codon is always AUG, or in fact, 99% of the time it's AUG, shown in red here. The light turquoisey blue is the anticodon region of this transfer RNA molecule, and it will always carry methionine. Although in fact, in the prokaryotic world, that methionine is a little modified methionine. It has an extra functional group on it called a formyl group. So it's F-methionine or formyl methionine. So here's methionine, and it's being held by this tRNA, and that is base paired to the messenger RNA at the start codon region. Then what happens is the large subunit attaches. So the large subunit cannot attach until this entire structure is formed. When this entire structure has formed, the large subunit will attach, and then you have an intact ribosome, and that results in the formation of these three pockets I mentioned earlier, the A site, the P site, and the E site. And you'll notice that this methionine is at the P site, so it's sitting there ready to go. And at this stage, initiation is complete. So the beginning is complete, and the ribosome is now ready to keep traveling to the right in this figure, down towards the 3' end of the messenger RNA. There's an empty A site. The next thing that's going to happen is a tRNA molecule is going to enter here. But that's going to be elongation or extension phase. So during elongation or extension of translation, this should say translation here. Let's just fix it. Okay, elongation stage of translation. Now that translocation word is a word that we will get back to. Okay, so the basics of elongation are a tRNA molecule that is carrying an amino acid, which is actually referred to as an aminoacyl tRNA. That means tRNA carrying an amino acid will enter at the A site, which was over to the right on the previous figure. A peptide bond will form between two amino acids, the one that is at the P site and the one that's at the A site. And then the ribosome will translocate, which means move one codon in the 3' direction. Then what will happen is the discharged tRNA, which means it's lost its amino acid, will leave from the E site. Those are the basic things happening during elongation. Here's a very bright cartoon showing that at work. Okay, so here's a ribosome, right? Here is methionine or formylmethionine at the P site. So initiation just happened. This tRNA molecule entered, and it has the appropriate anticodon to base pair with the codon of messenger RNA that is at the A site. It is carrying phenylalanine. These two amino acids are side by side, and that allows the ribosome, through the action of one of the rRNA molecules there, to form this peptide bond. And that's what this is all about, right? Peptide bond formation, building a polypeptide. As soon as that happens, the ribosome translocates one step. These get pulled to the left in this figure, or 5', and so now this phenylalanine carrying tRNA is at the P site. The tRNA that's at the E site has lost its amino acid. See, this is separated, and then this creates an empty pocket at the A site. This messenger RNA codon will only base pair with the appropriate tRNA anticodon. So in comes the appropriate tRNA anticodon. How does that happen? Because literally the ribosomes are swimming in a pool of these molecules. Okay, there are a lot of them. It's a concentration-dependent process. So others will try to enter, but they will not base pair. Only this one will base pair, and it's bringing the amino acid leucine. So in it comes, peptide bond is going to form, and then this is going to repeat. And meanwhile, the ribosome is going to travel one codon at a time towards the 3' end of the messenger RNA molecule. This is the direction to the right of ribosomal movement. And that will continue until you get to the end. Okay? So I want to again remind you that this is about peptide bond formation. So here's a cartoon showing a little bit more chemical detail. But here in blue is the ribosome, an intact ribosome, small subunit, large subunit. Okay? Here's the messenger RNA showing the codons. Here are tRNA molecules at the A and the P site, okay? And so what we're seeing is the actual full amino acid here, right? There's the little carboxyl group, there's the central carbon, there's the functional group that is on this, this particular amino acid, and there is another carboxyl group here. So here we've got a neighboring amino acid. There's the amino group. There's the central carbon. There's its R group. Here's its carboxyl group. Here's its hydrogen. A peptide bond is going to form between this carbon and this nitrogen atom. And there's the peptide bond there. Okay, so that's peptide bond formation. It's peptidyl transferase activity, and it's happening during the elongation or extension stage of translation. Okay, so then at some point in time, and usually fairly quickly, because this is a process that happens very, very quickly, you're going to get to the end of the messenger RNA molecule. How does the ribosome know? And the ribosome knows because it will encounter a stop codon. And when it encounters a stop codon, and here's UAU, this is one of the stop codons that exists in nature, what will happen is instead of having another amino acid enter, a set of proteins that are just generically referred to as release factor, RF for release factor, will enter at the A site. So here's the A site, the P site, and the E site of this ribosome. And instead of having tRNAs at the A site, we've got release factor, complexed with an energy-related molecule called guanine triphosphate, or GTP, which is going to energize this process. That will trigger the release of the polypeptide. Each of these yellow balls is supposed to be an amino acid. And then the ribosome will detach from the messenger RNA molecule, and it will actually separate. So it goes back to being a large and a small subunit separated. Okay, so another factoid, now that we've talked about the initiation, the extension or elongation, and the termination stages of translation, here's a fun factoid. This is still a really active area of research. And what I mean by this is the act of translation and the structure and function of ribosomes. So there was a Nobel Prize awarded in chemistry just a few years ago in 2009 to Venkatraman Ramakrishnan, Thomas Steitz, and Ada Yonath, and they were awarded the Nobel Prize in Chemistry for studies of the structure and function of ribosomes. They came up with the most detailed image, much like this, of an intact working ribosome. So people are still actively figuring out all of this stuff. Another fun factoid. Let's see, this is like a little bit, let me adjust this figure here for you. There we go. So this is like my all-time science hero. I really admire his work. He is now deceased, sadly, died just a few years ago. But this is a fellow, his name is Dr. Carl Woese. And he was a real genius. He figured out how to determine the evolutionary history between all living things by studying ribosomal RNA. He focused chiefly on the 16S and the 18S ribosomal RNA molecules, and he knew that they were very similar in all creatures, but also there'd be subtle differences that would allow us to fingerprint them. And it was that work that allowed the construction of a 3-domain tree of life, which we now know accurately, or most accurately, represents all life on Earth here on the tips, showing their common, last universal common ancestor, and the first pool of cells here. Here are the eukaryotes. Here in purple and red are the prokaryotes, belonging to the domains bacteria and archaea. All of this was figured out because Carl Woese understood what ribosomes are and how they are structured, what their functions are, and that they are evolutionarily conserved enough to serve as a fingerprinting molecule. So there are a couple of fun factoids for you, and with that, that is the end of this video lecture set on translation.